Yes, sir. Now it is visible.
Now it is visible.
Only slideshow. Only
yeah,
go to slideshow. That's all.
Yeah,
but slides we can see.
Yes, yes. So, friends, I'm sorry. I think that was some issues. So I hope now this goes well. Yeah. So. Yeah. So the views expressed in this webinar by me are personal and that do not represent of that of the institute and employer. I would like to make this disclaimer. Then I just So friends, I was talking about the opportunity in translational research that whatever we do basic research, we can translate into the let us say applied one by actually using particular, you can say, themes. Like here I was talking about it offers a great opportunity to develop the phytopharmaceutical product, and the second one is nutraceutical. But first let me concentrate on this, which is first time in India that and in the world that this new category of drug has been introduced, which is called as phytopharmaceutical. So there is this really offers a paradigm shift. So as it is mentioned, the paradigm shift has evolved, identifying one cause of a disease and one chemical entity to correct that cause. The traditional system of medicines in many countries, cultures that has used extracts from natural sources has by and large been ignored. That you will agree with me. In many scientific organizations, disciplines, we have ignored this over the, let us say, last 3 or 4 decades, but especially during last 1 decade, again, there is a renewed interest in this field. So thus the traditional systems of medicines which used various natural sources and their extracts has not found use in modern medicine. And due to increasing debates and concerns over the years, the US FDA in 2005 finally recognized a class of medicines termed as botanicals, which are plant-derived natural extracts and complex mixtures of compounds. These botanical drugs are not necessarily purified chemical entities to treat a disease, but the same class of drug was also adopted in India, as I mentioned, as phytopharmaceuticals in 2015. Yet, till date, no clinical trial of any phytopharmaceutical has undertaken in India, but several of them are under now different stages of development, and soon we'll have actually a phytopharmaceutical product approved by our DCGI in near future. So one such example I gave last year also during one particular this webinar, that the clinical trial of the extracts from the plant called as Patal Garudi, which is botanically defined as Cocculus hirsutus, which are the aerial parts of this plant. It is a runner kind of plant. So all its aerial parts are being used actually in medicine. So this particular phytopharmaceutical product development spearheaded by Sun Pharma has begun to mitigate COVID-19, and this is led by ICGB, CSR laboratory in Delhi, and another CSR lab in Jammu, IIIM Jammu, Indian Institute of Integrative Medicine on academic side. And DCGI gave fast approval for the clinical trial of above phytopharmaceutical last June for treating the mild COVID-19 disease. And that was in 5th June 2020. And the company Sun Pharma and IIIM Jammu are working very closely to push this product into the market to treat the mild disease. That is what the first phytopharmaceutical example was given. And one of our students who is from our department is the part of this team with CSIR IIIM Jammu. So he shared that initially this product they were developed for dengue virus infection. And as we know, the let us say expression and the entry of dengue virus and the SARS-2 coronavirus, there is a similarity. And based on that, actually DCGI gave the promising results for treating dengue virus infection. The DCGI gave approval for conducting clinical trials against SARS-CoV-2 for this particular phytopharmaceutical. So basically this phytopharmaceutical product contains the standardized extracts, which are actually qualitatively and quantitatively estimated with 4 markers which are actually required to be contributing to the biological activity. And these 4 markers are actually the interesting class of alkaloids which are present in this plant, Coculus hirsutus, and hirsutin was the major alkaloid which is there in this plant that plays an important role, shows this antiviral activity. So this is actually a class of, you can say, drug, phytopharmaceutical, and the definition all you must be aware. Just I will tell in short that phytopharmaceutical drug is defined as a purified and standardized fraction with defined minimum 4 bioactive or phytochemical compounds that are qualitatively and quantitatively estimated of an extract of a medicinal plant or its part for internal or external use of human beings or animals for diagnosis, treatment, mitigation, or prevention of any disease or disorder, but does not include administration by parenteral route. It is to be mainly, let us say, administered by the oral route. And as you can see that generally what is desired here and a win-win situation will be that you have a standardized fraction, not extract, a fraction which is the enriched fraction, let us say an alkaloid-rich fraction or a saponin-rich fraction, that depending on the, let us say, the in vitro, let us say an in vivo preclinical data, we can define these 4 bioactive markers in this enriched fraction, and that we have to ensure that we get the, let us say, we have the optimized process to make this enriched fraction that will be converted into a drug product. So the active ingredient here, as we say API, here we call it as APPI, active phytopharmaceutical ingredient, that you need to define with respect to these 4 bioactive markers. And that is what actually the need of an hour in this developing phytopharmaceutical product. And we are going actually for the product for obesity in this particular area, as currently we are very close to developing this phytopharmaceutical product, as all preclinical studies have been completed from a very well-known plant, that is Muraya konigi, all you know, a curry leaf plant. So in southern India, our foods and many preparations are incomplete without the use of curry patta. So from that, we have actually developed the anti-obesity product from this particular plant, where we have quantitated 4 bioactive markers, which are the carbazole alkaloids, including mahanimbine, coenimbine, conigesine. These are the 3 main compounds which are contributing to the activity. So it is a win-win situation. It is not just one, but 2 more markers that are there in combination are helping to reduce the, let us say, triglyceride levels very significantly in animal studies in the high-fat fed, let us say, high-fat fed diet into mouse model that we have developed, and done the toxicity study also. The not regulatory one, but the 28-day toxicity study has been completed on this fraction. So friends, this is the opportunity with one example, without giving much detail as it is under patent, but this offers us very good opportunity to develop the formulation from this enriched fraction for oral use, may it be a tablet, may it be a capsule, or may it be even liquid oral formulation, depending upon the nature of your APPI. And this actually enriched fraction, you can formulate and actually have the patent for the process that actually enriches, let us say, this fraction or alkaloids, for example, I have mentioned, from the extract. Extract, as all you know, which is a very complex mixture, may contain from 100 to 300 compounds. So from that, we have to, let us say, downsize and reach to a fraction where we have 10 to 12 compounds. Out of that, 4 are very well defined qualitatively, quantitatively, as well as they are contributing to the efficacy of that particular fraction. That is the need of an hour, or that is a regulatory requirement for a phytopharmaceutical to be developed. And friends, Department of Biotechnology, then CSIR and ICMR have actually signed an MOU last November to develop these phytopharmaceutical products from the, let us say, important medicinal plants that have been described in Ayurveda. So this really actually gives us very good opportunity as a paradigm shift in the drug development as a new category of drug. So that is what one need to see. So data you have to generate on identification, authentication, source of plant used for extraction and fractionation. As I mentioned, the enrichment, that is fractionation, process for extraction and subsequent fractionation and purification. This is where you have a chance to file the process patent. And then with the help of all biological data, preclinical, you can go for the product patent as well. Then the pre-formulation, formulation details of the phytopharmaceutical drug, manufacturing process of the designed formulation, and its stability data. So all you need to generate this data, which are required actually to form the part of IND that you can actually do, and it will be regulated similarly as our conventional pharmaceutical drug product. That is what actually an important thing in this particular product development that offers us a great opportunity. One can have good collaborations and try to develop these phytopharmaceuticals where a lot of funding is available under Department of Biotechnology, Government of India, ICMR, and CSIR. So these are the three funding agencies which one has to look for if you want to apply for the research grant for developing the phytopharmaceutical product. So this, in my mind, it is, or according to me, a very good area where most of us, whatever discipline you are working with, medicinal chemistry or pharmaceutics or pharmacology, and even natural products, pharmacognosy, we have great opportunity. And make good teams, good collaborations, and you have very good opportunity to develop the phytopharmaceutical product. So if you want to go a little easier, easier product to be developed, then one should go for a nutraceutical product. So friends, nutraceuticals where the regulations are much, you can say, milder. There are not stringent regulations, and we have to apply to FSSAI, food, that is actually the authority, Food Standards and Development Authority under actually the Ministry of Health, where you can get your license within 3 to 6 months, depending upon the type of nutraceutical product you are developing, which is very express, fast approval you can get for developing a nutraceutical product from a nutrient or from a medicinal plant that has nutritive value. We have so many plants. Like we could have also gone for curry leaf nutraceutical product, but we wanted to explore in phytopharma as it offers a lot of funding and opportunities. So we have gone for phytopharmaceutical, but one can go for the nutraceutical product development also, depending on the what form you are developing, whether it is from the medicinal herb, whether it is from the food ingredients, whether it is from the essential phytochemicals, which are actually promoters of good health. We can define that, or let us say prebiotic, probiotic. We have different forms of these phytopharmaceuticals where we can have the opportunities to develop them as a nutraceutical product. So you have to follow FSSAI regulations 2016, where we have a lot of opportunities to develop this nutraceutical product as well, apart from this phytopharmaceutical product. This is what in my mind are the various avenues for research are available nowadays in pharma for all of us, whether in industry or whether in academia and research organizations. We have a lot of such opportunities, friends. So with this, if I can take some questions on this, then I will move towards another lecture on this innovative teaching and learning pedagogies that we can use in, let us say, our day-to-day teaching of whatever pharmaceutical field we are teaching it. So that is what I would say. So I think Dr. Smita or Dr. Phatak, we can tell participants if they have quick questions, or you would like to take it at the end. I'm open for both, whatever you can say.
So we will take the questions at the end. You can start with the technical session.
Okay, thank you. So
Yes.
that's fine with me as well. So this is the technical session. So I will now share this screen. This screen is visible now, all of you?
Yes, sir.
Yeah, so thank you. So friends, with this title, I would like to tell some lacunas in our teaching methodologies that we are going through. And all of us are teachers. All we try to actually innovate ourselves, try to bring newness in our teaching so that whatever we want to convey, deliver to the students, concept-based teaching, then research-based teaching is really need of an hour that actually inculcate research skills in our students, especially the MPharm and PhD students. So where I think most of my discussion will be there when we are talking about this research skills and teaching learning, we can definitely talk about our undergraduate students, the pharmacy students. With this, let us see this actually elements of innovative teaching. So research-based curricula we need to design, and this curricula should be as far as possible student-centered. It has inclusivity of diversity, and very important one, the flipped classroom teachings. As your college or institute is already the NAC approved and all that agencies, they demand now innovative teaching and learning methodology. So one such, I think, methodology is adopted by many institutions that we now come across over the last 2-3 years, the flipped classroom teaching. This is one such innovative teaching and learning methodology that friends we have to adopt, and cooperative learning so that students feel that they are involved in this learning process. And it is rightly said, learning is a continuous process for whether it is for students or even I believe for the teachers. So we should have this cooperative learning and involvement of more and more students in a class of, let us say, 50 or 100. We should see that most of the students there, let us say, participate in this learning endeavor during our interaction in the classrooms. So research-based curricula actually enables students to become independent learners. Research projects help develop and test students' subjects understanding and follow their curiosity and explore a topic of interest in detail. So it is better if we try to inculcate these habits from third year onwards, when students actually prepare for a lot of competitive exams for their postgraduate education or even for their education abroad. Then students also learn to communicate their research findings. This is very important. And we all understand the importance of communication skills. So whatever, you may be a good researcher, but if you are not able to communicate your findings well to the audience, the research has actually not that much importance. So this is very important as we are talking about this research-based curricula. And the students get the opportunity to communicate their work to a broader audience and explain the significance of their ideas, not only to the teachers, but also to the industry people and innovators. So student-centered teaching should be there. That actually means giving students responsibility for their own learning. They become independent thinkers by developing the strategies and confidence to learn by discovery. Implement a modular structure that enables students to have greater choice and flexibility within their discipline programs and gives them opportunity to access modules from other disciplines. And friends, again, here I would like to say pharmaceutical sciences is such a very interdisciplinary field, which actually has a lot of connections and let us say linkages with many basic disciplines of sciences. And that's why I believe it's actually offers a lot of flexibility in a way that actually is required or need of an hour when we talk about this innovative pedagogies. Even develop novel ways to work in partnership with students, like enable them to co-create innovative teaching practices, employ them as teaching assistants in classrooms. And I'm sure many of us that do with our PhD students. And also in online and digital education communities that we are all going through since last 1 year. Then inclusivity of diversity, creating equal opportunities for the students. Then recognizing and harnessing students' diverse cultural backgrounds. As we see in India, we have different cultures, different ethnic backgrounds from where we do get students. Like I experienced this a lot at NIPER, as we get students throughout India. So that itself is a challenge for me as a teacher, that whatever I try to speak, first of all, they should understand what I speak in the class, as they are coming from different parts of the country. So this diversity, and to cope and with this diversity, and how I can make my teaching to the students very innovative one, very cooperative one, and student feel involved. And designing a diverse range of teaching, learning, assessment approaches. So we should not only embark on only the test, class test, written test. We should also try to have the debates, try to have the, let us say, the test which is based on, let us say, the objectives, like the multiple choice questions. We should have this type of test more and more than writing the very elaborate subjective questions. So this is another way we can try to create interest in students, giving them the innovative examinations, that we have the debate among students on a particular given topic, who gives the better solution for a particular thing. Like I was talking about enrichment, like developing an enriched fraction in a laboratory from an extract. So we really have the groups in students who can give, let us say, the better enrichment protocol. So they will get certain, let us say, reward. So we recognize such, let us say, innovation by students by giving them small rewards, giving them, let us say, some cash prizes even at times, that really boost actually the morale of the students, and they participate more in such research-based learnings. That what my experience I want to share with all my friends. Then removing barriers to learning. Of course, that is the endeavor all we do all the time, regardless of language support student need. Using lecture capture and captioning, as well as by making comprehensive lecture notes available in advance for students, so that in class they don't write anything. They only pay attention what I'm speaking. They are actually involved in debates. They are involved in discussions. They are involved in lot of question answer sessions. So that is what actually is possible if we give them our lectures in advance, whether it is a PowerPoint lecture, okay, whether it is some concrete notes point wise on a particular topic, that we make available to the students in advance before they come to the class. They will prepare, so that I always do that. What I'm going to teach tomorrow, that topic I give them well in advance, 2-3 days before, and tell them, okay, you prepare this. I will ask a lot of questions on this before I tell what is the newness in this topic and what are the concepts, basic things they need to learn. So that actually create a lot of interest in students. And the students who are not talkative, they are introvert in the class, they also start to speak sometimes if we adopt these practices. And that is what actually we have to make inclusivity of the students in the classes all the time. And I'm sure this is challenging to all of us, but we can always make effort and try to improve on this. Then I mentioned about the flipped classroom teaching. So I was on some inspection in UGC for NAC for certain institution during pandemic, friends, in this January. So what I find, the institution has actually this flipped classroom teaching for their BPharm as well as MPharm students. So we are very happy to see that. And even at some institutions, they are not yet introduced. So this is another very innovative method nowadays people are adopting. So this is what we all should try and endeavor to introduce this flipped classroom teaching in our classes. And that requires actually some software, some technologies, and the platforms. No doubt it comes with some investment cost, but I'm sure many good institutions have liberal funding, so they can adopt these flipped classroom teachings. So flipping a class means now designing lessons such that students learn content listening to lectures at home. This strategy allows more active engagement with students in the classroom, because they have the material, they have studied at home what is going to be discussed tomorrow in the class. So they actually prepare themselves that through different assignments, let us say the small questionnaires that we can prepare and give them before they come to the class. So these flipped classes allow the ability to meet with students one-on-one basis more often and at a higher rate, the ability to have more group projects, problem-based learning, and more laboratory experiments. In addition, students are expected to go home to complete their assignments by using the knowledge they have again in the class through hands-on training and lab work projects. Friends, these two last points, unfortunately, we are not able to do due to pandemic, but I would like to tell at Naipur Mohali, we have called all the 600 students. We have completely now masters and PhD together. We have 600 students on campus, but it's really risky now as the situation is worsening, and with actually much displeasure, we have to tell the master students to go back home again. So, but where I think these two components are only possible online, not offline. More and more lab-based teachings, that is only possible if students are here. But now many of us have to rely on making videos, making small clips of and demonstrations, hands-on training sessions on instruments. All that is need of an hour, that virtual teachings that we have to adapt to, friends. So this is another challenge in a way we have to face when we are talking about this innovative teaching and learning methodologies as well. So this is actually in a nutshell, you will find the classroom teaching, flipped classroom teaching model, where in class, during the class, student practice applying key concepts with feedback that is given. You can make small groups of 2 and 3 in the classroom, like we sit in cafeterias in 2 and 3 groups. We can make such arrangements in a class, and they have all these actually teaching aids available to them. Online teaching mode where internet based access is also given to find out the current concept if required during this classroom that students do. So students prepare to participate in classroom activities before they come to the class, out of the class, before they come to the class, and students check their understandings and extend their learning after this. Actually, the classes are over. That these are the main goals that we have to design during this flipped classroom teachings. Then cooperative learning, actually it is positive interdependence, individual accountability, group processing, interpersonal social skills, and the promoting the interactions. So all these are parts of our vital components of cooperative learning. Then innovative teaching learning methodologies, as I have mentioned, that we have flipped classroom, technology enhanced learning, mind maps, flowcharts. So we know that we teach a lot of instrumentation through flowcharts. But apart from instrumentation, we have to also make an effort that certain concepts in whatever subject, whether it is a lead optimization in medicinal chemistry, or let us say the proof of concept of efficacy in pharmacology, or let's say in natural products, that the structure activity relationship, the phytochemical constituents, and let us say the active constituents. All these informations that we can share to the students through the flowcharts. So it becomes more easy for them to understand, and even we can make certain mind maps as well for the students, so that can be displayed in the classes. So friends, again, this is all possible in offline teaching, but flowcharts we can also display in the virtual teaching modes. So even mind maps also. So that is what we have to make our efforts and try to, let us say, implement these methodologies in our teachings. So technology enhanced teachings, learnings here, actually digital and online innovations can be used to support and enhance more interactive ways of teaching. Online and digital technology can replace lectures and classrooms. Use of technology can further enhance our education, including real-time, two-way feedback system. The use of blended learning can be used to free up time in the classroom for more interactivity. Students can be asked to study online materials before coming to the class and will be better prepared for actual interactive classroom teaching. And here we want to give a lot of emphasis that our class teaching should be more interactive. It should not so happen that teacher is only speaking, students are only listening. So in between, we have to actually provoke students to ask questions regarding the basic concept and the application part that a particular topic we are discussing. So that actually lead to more interactive classroom teaching and learning for students. It allows to create international classrooms. And now by this, let us say, pandemic also has taught us we can invite any person. As we are teaching, I am sitting in my office. Without affecting my work, I have to only spend 2 hours and I can be available to the people outside, let us say any part of the world, not only within India, outside India. Even I can be reached to the students if my lectures are useful for them. So I can share my lectures with them online. So this is what, friends, another opportunity this pandemic has given. We can have international classroom. We can invite any expert in a particular area if he or she agrees, and we can have these interactions. So this is what another advantage of this pandemic. We have to make use of it in our teachings. Then mind map. Mind map is a diagram used to visually organize the information. For example, drawing information in diagrams instead of writing them in sentences. Mind map can be drawn by hand, either as rough notes during a lecture, meeting, or a planning session. These pictorial methods record knowledge in much easier way. Obviously, that we see nowadays in school learnings also. Even what I see in this ICSE, CBSE, we have a lot of such pictorial methods of teaching, which actually helps students to gain the knowledge and concept in a faster way. That than what we used to learn in our days, all of us, when we had our education in 80s and 90s. So the non-linear nature of mind maps makes it easy to link and cross-reference different elements of the map. Mind maps are much quicker to make and easier to remember. So for example, this one, possible mechanism of action of antibiotics as an antibacterial, that we teach in a medicinal chemistry class, for example. So inhibition of cell wall biosynthesis, here I need not to tell the different targets, different stages of this, let us say, bacterial synthesis is being shown, and different stages we can explore as the drug targets, as it is mentioned there with their mechanism of action. So with this, we can teach them different classes of drugs depending upon the stage of the growth inside the cell. We can do this particular and explain the students the mechanism of action of particular antibiotic or antibacterial agent that is being discussed class-wise. So this is a kind of mind maps one can show depending upon the topic that we are preparing and then teaching. Now, friends, apart from these methods, I would like to little bit talk about our institutions that are growing at very faster rate. Till the pandemic last year, we have seen the number of institutions have grown out of proportions. The following in particular have not grown proportionately: the quantity of quality teachers and training tools, quality of student training, quality of research output. This is all fastly actually disappearing with the large number of institutions that are being approved over the last 1 decade or so. So pharmacy education today is thoroughly commercialized and is in the hands of market forces to some extent. So that is another thing we are seeing. Then the private sector, which accounted for about 10% of students admitted in the 1980s, now accounts for 91% of all pharmacy students admitted in general. And the experts speak, our university system has lost its primacy and now beset by cross commercialization. Its autonomy has been eroded and undergrad education is undermined. So the autonomy of many institutions are taken away with this actually explosion of education that we see. And these are the views expressed by Yashpal ex-chairman, University Grants Commission, and he was also part of several actually new organizations that government is talking about since last 4 or 5 years. Like HERA, we are hearing a new education authority will come in place of UGC, but it has not come up as on date as we are speaking. But that is what we are thinking actually, to revolutionize our education system in India. Then very well applies to the most of the pharmacy education here, when we are talking about the autonomy is eroded with most of the institution. Overemphasis and spending on buildings by managements of private institutes and less on education quality. So I really don't want to undermine very good institutions. Okay, by saying this is a general opinion I'm giving. My intention is not to harm any private institution. We do have very good private institutions, like one we are here today, Modern College of Pharmacy, or I would say many private institutions like Amity. Like friends, I'm sitting here just 4 kilometers from Naipur Mohali. We have a new campus opened by Amity. Now, lot of scope. You see, Amity, we all know, a very quality education providing organization in private, and they have opened up and just very near from us. So we do have few calls from Amity, like our let us say the visiting faculty, like I got an offer. So friends, by saying this, I am not undermining. Just now I have given you in Mohali, in the knowledge IT city, this Amity has come on 60 acres. So many good institutions have come and they are hiring very good faculty now. So that is what I meant to say. They have to hire very good faculty so that the quality of education is not compromised. That is being imparted to our students. Then type of institution on quality scale. So to talk about my institutes, Naipur, Mohali, SS Nagar, as we say, we have highly qualified faculty. Every one of us has training abroad. And that is what the basic prerequisite in 1994, when this Naipur Mohali was actually started. It was emphasized by the mentors of this institute and by the, let us say, all top class academic personalities, as emphasized, to have high quality faculty, interdisciplinary training to them, best of the facilities we make available for the faculty. And that is why it is most favored by the students when it comes to taking admission for PG and PhD courses. Old leading institutions, they are old and established leading institutions. They have contributed majorly to the growth of our profession. Scope of further upgradation of facilities could be there in this old leading institutions. Then other institutions of respectable quality, upgradation can convert them into leading institutions, and local institutions that only meet basic AICTE PC requirements. Here, low-level institutions responsible actually to some extent for downgrading the quality of pharma education and profession. So that is what has been said by certain experts over the last 2 decades or so when we are talking about these institutions. So good pharmaceutical companies are awarding unskilled pharmacy graduates and postgraduates and have started preferring students from basic sciences, as all we are struggling from our, let us say, college days, that BSc and MSc were given equal opportunities as that of B.Pharm and M.Pharm students in industry. So that is what I meant to say here. A B.Pharm student has been considered as a cheap labor, and so is going to happen soon with the postgraduates, and it has already happened with the postgraduates already as we are speaking. So the problem is going to become more acute in the future if there is further increase in seats by AICTE without proper planning and consultation. And now we know there is less and less role of AICTE and more role of PCR. So that will actually strengthen, and our dual, let us say, regulation in pharmacy institutions will end soon. We all hope that. So unfortunately, in India, we have a dual statutory system, just I have mentioned, which is going out of control. Then the uncontrolled growth of institution is likely to become steady, as those established with sole objective of making quick buck will close under the influence of market forces. and the rule of survival of the fittest will eventually prevail. And so not much worry about or on this account. So the unfortunate situation that a pharmacy graduate is treated as cheap labor by employers must be amended. Academic institutions are expected to a big role in this direction so that our graduates are not treated as cheap labor, but as a skilled workers and well-trained pharmacy graduates who can really contribute in a bigger way in the growth of pharma industry. Change of motto of academic institution from excellent building to excellent teachings and training infrastructure with greater emphasis on modern instruments, attracting and hiring best faculty, continuous high faculty development programs, and that is what we are together, friends, that we are discussing how to actually enhance the quality of our faculty members through such actually training programs that we discuss and try to let us this platform to highlight what are the difficulties that we teachers face when a lot of principals, directors, or our management. So excellent academic buildings are beautiful only if they are equipped from inside as well. So this is what we need to see. So this setup generally, friends, we see in industry, in a quality control department or in R&D or the in-process quality control labs. We see all this, but where are such facilities when it comes to our academic institutions? I do of funding. Even want to buy, let us say benchtop, let us say FTIR instrument. It is not easy, friends, but we do have a lot of now schemes by government. We can apply under FIS programs, under DST to get our facilities upgraded with respect to the instrumentation that we are talking about. So we should have this all advanced instrumentations available to us so that our students get not only theoretical training, but they get the practical hands-on training, be it a HPLC instrument, be it a, let us say, GC instrument, be it a hyphenated technique like GCMS or LCMS. All this is very important nowadays in pharma industry, that our students are trained not only in instrument handling, but also the interpretation part where our students are lacking. So this is what need of an hour. By having these well-equipped laboratories, we need to do a lot of effort and give our best to give them practical hands-on training and interpretation of the data that is generated out of these instruments. So the teaching shall be concept-oriented and not only bookish, as followed by most of the pharmacy teachers. But I'm sure, friends, all of we are changing now, and we have actually adopted, let us say, a concept-based or research-based teaching in our approach. And curriculum must be interdisciplinary in line with global trend, and that is what I earlier mentioned. Friends, our pharmaceutical sciences has linkages, and it is very interdisciplinary science. It has linkages with many advanced branches of science, like you see, let it be a molecular biology or microbiology. Friends, with my collaboration in IMTech Chandigarh, I have learned a lot in microbiology, which I could not get chance to learn during my B.Pharm days or M.Pharm days. So that gives me an opportunity to have the linkages, interdisciplinary. So we have actually finding the bacterial efflux pump inhibitors as one of the solutions to the ever-growing problem of AMR, antimicrobial resistance. It is a big problem throughout the world which is being faced. So we can only understand by having good collaborations with microbiology experts. So friends, that is what we need to do. A lot of collaborations with different disciplines. And that's why I would always say pharmaceutical sciences at the center has a lot of scope for this interdisciplinary sciences collaboration that actually allows us to learn many new things over our, let us say, development of academic and research careers. So also it must be designed with futuristic vision involving all the stakeholders. So stakeholders of pharma education in India, central government, policy, framing of policy, approval for establishment of central institutes, national institutes like NIPERS, as we are talking about, statutory bodies like AICT, PCI, we just discussed. They are regulating the education within the pharma. State governments, policy, approval for establishment of state universities, local institutes, etc. Then various organizations, UGC, DST, DBT, CSIR, and so on, ICMR, Ayush, and many more. So funding of research and fellowships to be actually now available more and more to all of us. We can apply for that. Then imparting of education by universities, institutes, colleges, and not only that, industries, CROs, CRMs, hospitals, clinics, and CDSCO regulatory authorities, bodies in our countries. They are also part of actually these stakeholders of education, as they also offer us a lot of employment to our students, and several professional bodies like IPA, IDMA, then IPGA, APTI, as we are talking about, and many more. And students are actually the big stakeholder, and they are receiver of the education, as we are discussing. So with these friends, just I will take another 10 minutes to just quickly go through applying this QBD to pharmacy education. All we know, this is another concept, which is an old concept, friends, as all we know, but that is being now adopted and applied in pharma recently. That's why we talk about QBD is a recent, but QBD is there since long time in automobile industry, from where this concept has come, then aeronautical industry, and now we are, because there is a lot of risk involved in automobile and the aircraft industry. So now that we have realized in pharma also, there is a risk involved, and risk assessment is an integral part of QBD, as all we know, Q10 guidelines there. So if all our teachers, you must have taken many such courses over last 5 years on QBD. And I was instrumental here under this, our high-tech programs under Ministry of Health and Family Welfare. Many foreigners, they have taken this advantage of QBD programs that were imparted by NIPER in the last, especially 5 years, where I was course coordinator. And I have learned a lot myself, doing research in natural products, medicinal chemistry. I have learned a lot, and I can use this actually QBD in my chemometrics that I am using day to day in the analysis. So that is why, that is how I look. Even this concept of QBD is very important in our pharma research. Similarly, it is important in pharma education also. That linkage I would like to share here. So QBD is a new concept applicable to pharmaceutical development and production. And this particular issue you must have read, all of you, August 2014 Pharma Times, QBD pharma development, where many articles, including articles written on application of this, let us say, QBD in even phytochemical analysis, apart from the, let us say, the formulations and APIs. So it is rather a revolutionary change for assurance of product quality. So we have to move towards quality by testing, towards the quality by design. So QBT, quality control by inspection and testing of finished product, is now transformed into QBD. So quality built into a finished product, and it is not merely tested, but it is built at every stage, starting with the raw material to the finished product. That is what the concept of QBD we are talking about. So, and PAT is very important aspect of this QBD, or a very important element of QBD, that is process analytical tools. So that online, during, let us say, the product cycle, we can assess, we can build the quality, and also check the quality in the product by using these PAT tools. So for example, IR. So near IR is very, fantastic technique in material characterization. So where we can use it in raw material testing in different ways, as we can use IR. So that is NIR in material science, people are using this a lot. Then dry compaction, wet granulation, FBD milling. So we have online particle size analysis, even NIR, loss on drying methodologies, online UV cleaning, and methodologies on online blend uniformity, then compression also inspect with NIR core potential, NIR coating thickness in the coating process, and imaging of blisters. So you see, friends, here NIR has emerged as a very important PAT tool during this QBD over the last 2 decades now already. Now the key elements of QBD, all you know, I will not go into detail. We have to define target product quality profile, then the design product and manufacturing process, identify CQAs, critical quality attributes, process parameters, and sources of errors, control manufacturing processes to produce consistent quality over the time. So QBD is talking about different spaces: knowledge space, design space, and control space. So extension of then QBD to education. So quality by testing in education to quality by design in education. That means instead of only conducting tests like in the semester end examination or annual examination pattern we have to move towards quality by design in education by conducting periodical tests, small tests, tutorials, and similar kind of tests that we have introduced in assessing the knowledge that is being imparted to the students. So QBT, quality through testing and examination, is moved towards QBD, building quality in students to effectively design policies, curriculum, and teaching, training programs, incorporating continuous education, imparting continuous education to the students. So expected teaching quality, young students want solid education where curriculum and teaching methods are up to date. They want teaching and training that helps them adapt to the reality of this new millennia and is useful for entering active life, the world of work, and society, which is becoming extremely competitive day by day. That all we are experiencing. Therefore, the objective of teaching and training shall not just be on increasing the key qualifications of the students, as it is practically happening with mushrooming of colleges and uncontrolled increase in number of seats in pharmacy institutions, where only degrees are distributed with minimum emphasis on knowledge and skill development. And QBD, a teaching and training program that aims at all students of a class having similar knowledge and skill set as assessed through a continuous evaluation program. So despite that, students of a class, maybe with different backgrounds or levels of intelligence and capacity to learn, the students are given certificates of completion of the course and degree at the convocation. So end examinations or end term examinations not necessary or given very low weightage due to continuous evaluation in the current education and examination patterns. So the continuous and comprehensive evaluation pattern of CBSE is designed to reduce the stress on students. This is very important. So earlier final examinations at the end of the year, but which is 100% weightage was given, but now 2 summative assessments are there where weightage is now 70 and 30% for this SA1 and SA2. So that means, friends, the continuous and comprehensive evaluation pattern we have to adopt in our education system. And a great advantage of this is freedom from anxiety and stress from the test and examination among our students that we see actually. And as all you agree, all this has also led, pandemic has led to the open book examinations. And now this is more and more liked by the students. They are preferring for this. What I have observed at NIPER also during last 6 months or so through this online teaching. So an extremely stressful situations are created sometimes at some places like Bangkok University makes students wear anti-cheating helmets during midterm examination there, which is very, I was very shocked to see such type of things. At least in India, we don't have this. But this could be actually a stressful situation, which is, let us say, imposed on students. So as such, also in today's environment of campus placements, the employers hardly pay attention to mark sheets. And I totally agree this, what I see in our campus placements. let us say the company people who come HR or the technical people from particular industry, they see the knowledge level, they see the communication skill, they see the confidence, body language in the students, than merely looking at whether he has scored 90% or 95% or A grade or A plus grade. So friends, these do not matter much, and students who are smart enough, who knows how to present him or herself, gets the job than the studious students having 90% marks. That we have seen. But at the same time, the campus placements are reduced now due to this pandemic. At the same time, this is a challenge what we see, but we are doing this online placement for our students now. And over the years, what I have seen, I'm involved as a placement coordinator for my department, apart from the central placement cell at NIPER, and this is really challenging in time. What I see, the placement over the years have dropped. This is another worry part, and students take admission to the good institutions with the hope that they get better campus placement. And this is a scenario with NIPER Mohali. So friends, what I want to tell. So apart from the bookish knowledge, we have to make students able and ready to take the challenges in pharma industry, where they are able to solve the practical problems. And that is why I say we have to give more importance on concept-based teaching and inculcating the skills among the students than only evaluating the answer sheets and giving them the marks. So this is what I would like to say also as my personal experience with all of you. So the expected QBD elements in short, at macro level, you see these were the 4 elements we have seen. So define the product quality profile. So pharmacy students with knowledge and skill suitable for job. I think somebody's mic is actually on. Please mute it. Some disturbance we are having. So sorry. Then the design, product and manufacturing process. So that you can link to the robust education policies, high quality institution, high quality infrastructure, high quality curriculum. The curriculum being, let us say, revised, let us say, definitely every, not less, but every 5 years minimum. Even now it is the time need of an hour is that we revise in 3 years, the curriculums. And even we have more and more, you can say, these faculty development programs for our faculty. That is the design, product and manufacturing process. You can link this to this particular parameter of QBD, CQUS, that is critical quality attributes, good employability for each and every student. And that is what I mentioned about the campus placements, control type and total number of institutions, quality of faculty and student intake number. So this is the onus on every institute's management and their top thinkers, okay, how to actually control this quality of faculty and the student intake number, so that this is not jeopardized, and at the same time we impart the quality education to the students. So at micro level, you see here the target product quality profile, student whom any let us say employer would like to hire without a blink. That should be the repo or brand image we need to develop with our institution. Robust institutional teaching and training program we have, and they are being continuously upgraded. CQ is equal learning of all students, identify all variables that affect the learning of the students, control on all variables through efficient mechanisms. So that is how, friends, QBD elements can be applied in education, and that is what I have mentioned about QBDE. So the steps are, you have to build knowledge space, assess expectation and annual job vacancies in industries, CROs, CRM, CDSCO, state regulatory, government and private testing laboratories, not only industry, also hospitals, modern chain of pharmacies, individual retail outlets as well for that matter. Study education and research model of a leading Indian and international institution list all variables that can influence equal training of students in a class. Then we have to build a knowledge space, build design space, use optimal statistics to assess the requirement of the number of institutions, infrastructure, faculty, and students. Use best statistical and design tools. Use design models to outline most suited curricula. So here, friend, at point number 1 and 2, we are using DOE, design of experiments, as the main element here in the design space. Then apply design of experiments, that is DOE, to identify variables which require stringent control, and build control space, controls that can be implemented by regulatory agencies and also educational institutes to radically change the current scenario of pharmacy education. So burden on the students, that we all know, lot of burden, lot of curricula is being bombarded on our students nowadays. So some students have expressed on the Facebook in a few years back, actually, in a way it is true, but there is no option also, I would say, at the same time. But this is the, let us say, feeling of the students. Like one can say, समंदर जितना syllabus है, नदी जितना पढ़ पाते हैं, बाल्टी जितना याद होता है, गिलास भर लिख पाते हैं, चुल्लू भर नंबर आते हैं, उसी में डूबकर मर जाते हैं, डूब जाते हैं. But see, this is what actually the sentiments. We should not go by that, because we cannot sometimes decrease the syllabus in certain disciplines. But that is what the sentiments of students. Sometimes we have to take care, consider it, and be genuine to the students. Nowadays, believe me, the student life we had and student life, all you will agree with me, the current students is totally different. There is again a vast change here in the students' attitude. They don't want to learn. They don't want to have a vast syllabus. They want to have a very small syllabus and lot of knowledge and placements. That is what nowadays students want, and they don't want to put hard work in the laboratories also, as we teachers wish. So this is another challenge as a teacher I'm facing. I thought of sharing with you nowadays, especially with the M.Pharm students. That is my actual experience. Having said all this, the sentiments of the students, and it is a scientific tool for making education and learning of fun, and that is again another challenge, friends, we all teachers face, how to make learning very exciting and enjoyable exercise, and at the same time it gives a lot of fun in learning to the students. So this is all very easy saying than done, friends. So with this, just at the curriculum, how we have designed, how it is different. I'm talking about pharmacognosy natural product as I'm dealing with. This is the PCI syllabus. M.Pharm, I think regulation 2000, I guess 14, if I'm correct, or 16. M.Pharm regulation 2016 for PCI. This is pharmacognosy syllabus, where I see it's quite good syllabus, but some emphasis is lacking. So emphasis that gives students more flexibility, and let us say more interdisciplinary nature is missing here, because there is no linkages with medicinal chemistry, more or pharmacology, or even for that matter, the platform technologies, drug delivery systems. There is no emphasis in this syllabus, which is definitely required 1 or 2 courses. That is what I believe when I see even our syllabus. And our syllabus, we design more emphasis on chemistry in natural products, because that gives also a lot of training to the students, and in a way make them enable to compete with placement opportunities with medicinal chemistry and pharmaceutical students. That is, I believe, and accordingly, you see, that is our curriculum. In semester 1, we have kept it general. But in semester 2, we have made it very, in detail, and giving more emphasis on the chemistry aspect, which is totally lacking in the natural products pharmacognosy students. Because unless and until you know chemistry better, you will not able to pinpoint what are the lead molecules. Friends, I always believe all the medicinal chemistry that we see today, it is developed based on natural products only. It has developed from or originated from natural products only. So we need to tell our students that the lead molecules coming out of nature, They are more drug likely properties, they are more druggable and than the synthetically generated compounds in laboratory. So that's why we have to make our curriculum more chemistry oriented in when it comes to the natural products and pharmacognosy. That is my thinking, and that is what we try to do. And all new NIPERs that have come up, apart from NIPER Mohali, we have 6 more NIPERs across the country in different parts. They have all adopted NIPER Mohali model. In the curriculum, all NIPERs are different. All NIPERs are autonomous bodies under the Department of Pharmaceuticals, Ministry of Chemicals and Fertilizers, Government of India. So I thought it is important to tell to you friends, because most of your students, they actually compete and get admitted in our institutions. And I do get a lot of students in NIPER Mohali from Maharashtra, especially from Pune, then Nasik, and then even various other parts of Maharashtra, Aurangabad, even Marathwada. We see now more and more students are coming to our institute, which was not there earlier. And Pune, Mumbai, actually they are there anyway. So that is how we can actually attract more and more students by developing the innovative curriculum that gives more opportunities for the students to learn. And at the same time, they are exposed to a lot of experimentation. It's not only teaching, they are also given the practicals individually. They are being told to isolate a natural product, purify it, characterize it, a complete practical. So that goes for, let us say, 8 weeks. Student has to continuously do. Sometimes they take actually even 3 months to accomplish. So that is that sort of training exposure we need to have for the students. And we do have the formulation component, applying the conventional formulation strategies as well as novel drug delivery systems to the herbal drugs that we teach in our courses at our Department of Natural Products, which is different than any other place or different than several other places where MPharm pharmacognosy curriculum is being delivered. And then I have developed this course in my institute in the department, and we do get now a lot of placement opportunities in CCRS, in under the Ministry of Ayush. Now Ayush has come up with big ways, and they have many scientific and technical officer posts, and they are taking out traditional medicine students. Earlier, initial 10 years, we had a lot of problems for placement to these students. But now, friends, the scenario has changed as more and more emphasis is given by Ayush Ministry. Now, all we know since 2015, we have a separate ministry for Ayush, which was only a department earlier under Ministry of Health and Family Welfare. So that actually, now we see this course was very useful, that we thought with this vision that it will create more job opportunities, and we have a different training to the students from the same department under these traditional medicine programs. So this is another discipline we have developed, which is not there, I think, in most of the places in India under pharmacy, let us say, institutions. So this is what all about the flow charts that we have to use, I have mentioned. So friends, I think due to the shortage of time, I will not go into detail, but when I was talking about the enrichment fraction, I will tell with this example. We have prepared a boravinone enriched fraction or rotenoid enriched fraction from this very important medicinal plant called as Punarnava in Ayurveda. A small example, 5 minutes I will give, and then I will end up my talk. I think I'm talking since last almost 1.5 hours. So it might be, let us say, lunchtime to most of you. So I will just wrap up this example in 5 minutes to all of you. You must be knowing this plant called as Punarnava. There are monographs in our Indian Pharmacopoeia, National Pharmacopoeia on this plant in IP 2007, 10, 14 editions, and even in the edition it is there. So what this monograph tells actually, Punarnava contains not less than 0.005% weight by weight boravinone B calculated on dried basis. Now friends, this is a phytochemical reference standard, a marker, analytical marker for this plant, which is boravinone B, which is a rotenoid, a modified isoflavonoid compound that is present in this plant, okay, in the roots of this plant. So this is not available commercially. So how we do assess the quality of this particular herbal drug when it is used in many Ayurvedic and herbal formulations? So the way out is that we develop the fingerprint with the authentic raw material and compare that fingerprint with the sample material that you receive when you do the work on the roots of this plant. And simply this TLC or HPTLC fingerprint will be enough to assess the quality if phytochemical reference standard is not available. That is very well also mentioned in the Indian Pharmacopoeia by the Indian Pharmacopoeial Committee there. So this is where actually these monographs now finding more and more place in our national pharmacopoeia. And if you see the 2018 edition, we have almost 140 monographs on herbal drugs in Indian pharmacopoeia, and it is going to increase further. More and more monographs will be incorporated in our national pharmacopoeia. That tells us the, you can say, more and more popularization of herbal medicine and acceptability of common public in general. So this plant contains these compounds. I was talking about variety when we started this work. These actually 10 rotenoids, boravinones, were reported from roots of this plant. We have done detailed phytochemical investigation and prepared this enriched fraction actually. And that actually gave us very much good learning experience. That is what I will mention here. This is the flow chart. So friends, this is the enriched fraction one of my PhD student made when he worked on this plant. I told him, why don't we prepare the enriched fraction? You find the method, see the literature and see what you can do. So he come up with a simple protocol. You take 20 gram of the 70% ethanolic extracts, which is prepared from dried roots of Boravia diffusa. Then we have passed this extract on the diane HP20 resin, which is commercially available at very low cost. We can use this, and we have eluted starting with water with methanol further in gradient elution mode. 90% methanol fraction concentrated on rotary operator under vacuum. And what we see that most of these boravinones, what I have shown you here, these ones, rotenoids, they have been enriched in this fraction. That we have confirmed with HPLC analysis on detail, and 100% methanol has very less amount of these compounds. They do have, but majority of the amount of these rotenoids have come in this 90% methanol, and then it also had small amounts, so we have combined both this fraction, and that lead us or gave us rotenoid enriched fraction. And friends, that is need of an hour when I talked about the phytopharmaceutical product development, that we try and endeavor to prepare these simplified extracts, fractions from the complex extracts. That actually offers a lot of opportunity to do a lot of good research on in this area. So this rotenoid rich fraction then we have utilized for the formulation development, like we have gone for phospholipid complex formulations, or even the, let us say, liposome formulations you can prepare from these enriched fractions, which are prepared from the complex extracts of a medicinal plant. So for example, and this I can show here. So you see the enrichment, 5 mg per ml, 10 microliter injected to HPLC. This is the HPLC chromatogram of an extract, 70% ethanolic extract, and you see the absorbance, 0.6 to 3. So we can clearly see enrichment of these rotenoids. All these peaks here, they disappeared. They are very negligibly present, and more enrichment of this. And friends, this peak number 3, what I labeled, we have done LCMS analysis and identified before isolation this as boravinone B. This is the, you remember, phytochemical reference standard mentioned in IP. So this is the compound that we had isolated in gram quantity from this plant that is available with us as a phytochemical reference standard, boravinone B, by my PhD student, who also got this Farm Innova innovative PhD thesis award. One of his research project was on this Borrelia diffusa there. And we have, let us say, formulated this into a phospholipid complex formulation that clearly gave advantage over the conventional ethanolic extract when we have studied its biological activity and done the oral pharmacokinetic studies, bioavailability studies in rats. We have found the advantage of formulating this enriched fraction into a formulation. So that is what more and more research opportunities are available in this area, which is actually very easy to do. The phytocomplex, phytosome formulation or phospholipid complex formulation, or if you know a certain phytoconstituents or phytochemically the bioactive constituents, so that we can take it for even the further advanced drug delivery systems like we have tried with. I will not go in details with the boswellic acids which are present in this Boswellia serrata. As all you know, it is salicylic Google that is very useful in arthritis. Lot of clinical trials confirm the efficacy of this Boswellia serrata extract in treating osteoarthritis. So the major bioactive constituents are this acetyl beta boswellic acid and 11-keto beta boswellic acid. So we have isolated this and formulated into a polymeric nano particulate formulation that clearly showed a lot of advantage over the conventional extract formulation. So these are the avenues that offers us in this area that we can do a lot of formulation research that we have done, and many more such formulations are being developed in my laboratory. Like I mentioned, we are working on this, let us say, curry leaf phytopharmaceutical product, which we are going to now finish the preclinical studies and apply for the, let us say, regulatory approvals with certain industry partner that we have just identified, so that we get some funding from DBT. At the same time, they help us in trying to commercialize with the industry partner that the product for obesity we want to develop from. Let us say that is a current ongoing work in my laboratory towards the product development from Muraya, Pune. Okay, that I want to tell. So these are the all, I mean, things. And over this course of action, we had isolated 5 new boravinones from the roots of Borrelia diffusa, and that actually helped us to publish this very good paper for my PhD student in the Journal of Natural Products after actually getting the process patent for the enriched fraction. So this is what the thing. Then similarly, we have prepared the triterpenoid enriched fraction from Terminalia Arjuna that is under publication in phytochemical analysis. As all we know, this is very important medicinal plant as cardiotonic in Ayurveda, and it has a lot of applications. Stem bark of Terminalia Arjuna, for example. And these are the 4 or 5 triterpenoids that we have prepared the enriched fraction from the 70% ethanolic extract or methanolic extract of stem bark of Terminalia Arjuna. And there is a monograph also on this plant in Indian Pharmacopoeia 2014 as well as 18. And these are all the HPLC chromatograms of the preparation of triterpenoid enriched fraction from the Terminalia Arjuna ethyl acetate extract. You see this is a complex extract, and you see this is the enriched fraction that all we removed. This particular, the condensed tannins which are present in a very high amounts in this extract, which are the, I can term them as procyanidins or catechins. Difference, they are present as we see them in green tea. They are also present in this extract of Terminalia Arjuna. So friends, thank you so much. This is the main gate of our institute. If many of you have visited, if not, I invite and welcome you to visit our NIPER at SS Nagar, Punjab, which is very near to Mohali Cricket Stadium. All you know, it is just 1 kilometer from the Mohali Cricket Stadium, our campus. This is our natural product field laboratory. I am very lucky to have this very extended facility to my department. We have this 3,000 square feet facility to do the process chemistry research and even the processing of herbal drugs at pilot scale. We can do it here in this field laboratory of the Department of Natural Products. So friends, I am sitting here in C block. First floor, second floor is natural products. Ground floor is pharmaceutical analysis. So I'm sitting here. This is my chamber, HOD room of our department here in the Department of Natural Products. So I thank you all. I know I have very less time left for question answers, but still I'm ready to take the queries, questions, whatever you have related to my talk of teaching methodologies, or let us say the innovations that we can do in natural product research and otherwise also. Thank you so much.
Sir, there is a question from Dr. Rohini
Yes,
Chauhan.
please.
How classroom teaching is a useful technique for graduate students?
How can you just pardon? Please,
How flipped classroom technique is one of the useful teaching technique for graduate students?
Yeah, graduate students, definitely, if the class is 100, it is difficult, I know, the class strength. So you have to distribute, it's a challenge, that class into smaller fragments. So one class you have to take 5 times, for example. If I'm a teacher there, I will make 4 or 5 groups, 25 each. And then in the groups, if I'm teaching them medicinal chemistry, a particular topic, so we'll give them the theme and tell them to see the basic chemistry, mechanism of action, structure, activity, relationship. Through this, let us say, charts, they will prepare, discuss among themselves in the groups, and then I will interact with them group wise. What you understood, ask them the questions, for example. This is how it can be useful. And if they want certain, let us say, I am talking about, let us say, antibiotics, any class you take. In that, just now I mentioned, we have a big challenge of AMR, antimicrobial resistance, for a particular group of antibiotics. What is that drug resistance is reported in literature. It may not be there in a textbook, all the details. So students can be given online access immediately right there. They can see, check if a particular group of aminoglycoside antibiotics, what is the current knowledge there on their resistance, what kind of resistance, what bacteria have shown resistance, and what are the determining factors of that resistance. If I can explain that to the students, once they check that, it will be more usable, understandable for the student to understand. So that way you have to actually design your topic for every lecture and make the groups. Actually, it has to be in the groups. It cannot be for the bigger classroom. Flipped classroom teaching is not useful. You have to divide that class into the, let us say, at least 25 to 30, and from that make 5 groups and tell the students, groups of 3, learn the particular topics. In the same topic, you can make subtopics and give them this target that you have to focus on mechanism of action. One group will focus on SAR, one group will focus on what is the current status, one group will focus on what are the commercially used drugs there, are there any pharmacovigilance data on those drugs for their safety. So if you combine all these aspects, definitely this will be useful. But it requires a lot of actually infrastructure and investment from management side. Then only we can use this teaching. These are new to us, many of us. I want to tell to all the fellow colleagues. It is there at very few institutions, even as on date, we are discussing and debating.
Okay, thank you, sir. Since we are falling short of time, we will take only one question and we will sum up here. Thank you, sir, for giving your valuable insight on avenues for research in medicinal and applied research. Sir highlighted different points like concept of phytopharmaceuticals, opportunities, products and nutraceuticals. In addition to it, during technical session, sir gave knowledge about innovative teaching, learning and research skills in natural products curriculum, which included basic teaching elements like research-based studying, student-centered learning, inclusivity of diversity, flipped classroom teaching. Then different innovative teaching learning methodologies were discussed, which included technology-enhanced learning, mind map, flowchart, etc. Thank you, sir, for
Thank you so
sharing
much.
for spending
Thank
your
you so much, and thank you that for the overwhelming response. As the participants, fellow colleagues, teachers are there. I hope this talk will be useful and motivating, inspiring to many of them. Then I will think that whatever time I spent has some meaning with all of you.
Thank you, sir. That's
Yeah.
it. Please
Thank you so much, and thank you, Dr. Chaudhary. It is really pleasure interacting with all of you. Thank you so much.
So much.
Thank you.
Sir, please kindly accept this virtual bouquet on behalf of convener, Dr. P.D. Chaudhary, Principal of P.E.S Modern College of Pharmacy, Nigdi, and Coordinator of STTP, Dr. Atul Phatak, Professor at P.E.S Modern College of Pharmacy, Nigdi, as a token of gratitude. So please
Thank you so much. Very well appreciated. And let me tell you one more thing. I really like the way you have inaugurated and had Saraswati Vandan. It's a really good concept. It was very refreshing at the beginning when we started. Thank you so much.
Kindly accept the certificate from
Sure.
Pharmacy, Nigdi, Pune, as a token of appreciation.
Thank you so much. Appreciated. Thank you so much.
Thank you very much.
Thank you, sir. Thank you.
Thank you.